All right. Yeah, today we have Julia with us, and Julia is talking about one of her very recent publications, in fact, which is, I think, however, a very also important one, basically on working memory or short-term memory, as it is called in more colloquial terms, and how we manage to process the information in it. So, what did you do in a nutshell? What did you find out?
So before we get to dig deep into this topic, I would like to give a short example about working memory that we all have in our daily routine, I would say. So imagine you need to remember a phone number, some digits, and people are talking next to you. So in this case, there are some processes in your working memory which are active. So for example, you need to maintain the digits in your working memory, but you also need to shield these information against distraction from the outside, like the talking people. Sometimes, of course, you also have to update your working memory when new important information is coming in. If you see, for example, that one digit is wrong or something like that. So, previous research assumes that our working memory has a gate, and this gate can be opened to let new information in or closed to shield against this other distractions to keep unwanted information out.
So there is already something around where you could hook on as a concept, let's say. But what exactly did you do as compared to other studies that are already out there?
Yeah. So first, we wanted to know if there are distinct neurosignatures which modulate this working memory management. And we also wanted to know if there are specific brain regions involved. And for the third one, which is really interesting, is we wanted also to know how these brain regions work together, interact to enable this working memory management.
So you think, say, that there are different sorts to communicate this information, maybe in different sorts of brain activity, and that also different brain regions ascribed to these different activity patterns all have to come together.
Yes.
So it's kind of a whole brain endeavor, and information is being transferred to different regions. And so, what exactly, how should we, let's say, imagine this to happen?
So first, of course, our experiment in the lab is not as complex as daily life,
Sure.
but it's a good approximation of that. So we used a working memory paradigm together with some computational modeling techniques. We can also call it mathematical modeling to analyze our experimental and EEG data, which we recorded simultaneously to that. And our goal was to investigate the underlying neural activity of working memory processes like this gate opening and gate closing. And for this, mathematical modulationing approach showed that information is not continuously let into working memory, but it's more like a piling up of information. And only when a threshold is reached, so enough information is available, the gate will open or close. And on the other hand, with the EEG analysis, showed that there are distinct neural signatures and also distinct brain regions involved in these processes. And the most interesting part I find is that we find specific connectivities or communication patterns between these regions. And we found, for example, here in the frontal region, in the parietal region, and also in the occipital region, but also here in the parahippocampal region, we found some activation, and these regions communicated with each other.
So you're talking about activation, communication between different brain regions, but what are these activities precisely? So what did you measure?
So when we speak of neurosignatures, we always speak about brain waves at specific frequencies. And these brain waves can be either a bit slower or a bit faster. For example, we found something in the beta frequency, which is a quite fast brain wave, which is considered to play an important role in maintenance or monitoring of working memory information, or also the reactivation of memory information. Then we have, on the other hand, the alpha activity, which is like kind of a medium fast frequency, which is considered to be a gatekeeper. So it decides which information comes in and which should be kept out.
Okay.
And the third one is the theta frequency, which is quite slow, and it's considered to play an important role in cognitive control. So when we want to ensure goal-directed behavior.
Right, I see. So you have, let's say, three different signatures of neural activity, and they have to interplay somehow. But how is this interplay organized exactly?
So we found that we had an observation that especially in the theta and the beta frequency, the communication was set between regions involving memory processes and action selection, and also regions that inhibit thoughts and actions. So we showed that the exchange of information across frequencies was simultaneously to the fact that we keep information in our working memory. And we showed also that alpha frequency is, for example, important in gate opening, but in totally different regions. So that's an important finding, because we showed that gate opening is not just the reversal of gate opening or of gate closing or another process, but it's two different distinct processes which take part across different frequencies and across different regions, which was not well understood before.
So it is not that you let's say open one and the same door or close one and the same door, but let's say keeping the gate open is one door and close it, or the mechanism to close it is, let's say, another mechanism behind. Yeah. So it is not a one-to-one or a reversal in these things, which I think is fascinating, because usually people think of, okay, what's functioning to let information in is also functioning to let the information out. Yeah, which is, however, not the case. But in how far does this really extend what's already known?
So the new thing about our study was that we did a combination of mathematical modeling and also cutting-edge EEG analysis with a clear concept about working memory from cognitive science. So oftentimes you have people using fancy methods, but the connection to this cognitive concept behind that is lacking. So we started with the cognitive concept, and we showed how to get the best of two worlds with our analysis. So from cognitive psychology and neuroscience. And I think this is the best way to really understand how the brain works or how the brain functions.
As is sort of, let's say, a psychological neuroscience, yeah, when you start from psychological terms and develop a psychological question into a neuroscience perspective, but not the other way around, yeah, because it provides you some more constraints. This is really, I think, innovative and excellent. But I mean, working memory is a very broad concept. Yeah, it has been researched since decades. Yeah, but in how far would you, let's say that what you find out may also be of relevance when it, let's say, comes to clinical research, clinical neuroscience research?
So I think integrating basic neuroscientific findings are important if you, for example, want to enhance diagnostic accuracy. So, with the understanding of distinct patterns in the theta alpha frequency band, it allows us, for example, to develop diagnostic markers for conditions where working memory is impaired, like you see, for example, in Alzheimer's disease or schizophrenia or also ADHD. And further, we could use these findings in the field of neurofeedback therapy, which we already use in our hospital to enhance concentration and attention in kids with ADHD. So, with this real time feedback on their brainwaves, individuals who suffer, for example, from working memory deficits could learn how to enhance their working memory performance. And this could alleviating symptoms, for example, also again in ADHD or, for example, also in my cognitive impairment.
Yeah, I think this is really excellent, because you start right away from a psychological concept, which you then translate into really nowadays neuroscience research. So it's kind of a psychological neuroscience where psychology informs the neuroscience steps to be taken that can then also be used in a clinical translation, which is, I think, really new to the field.
Yeah.
Yeah, that's really great. So thank you very much for coming.
Thank
Yeah,
you for
thank
having
you
me.
for having. Yeah, of course. And thank you for watching us. Bye bye.